Hi, and welcome to my OCRA A-Level Biology Revision session with me, Christine. So today's lesson, I want to look at regulation of your blood glucose concentration, and that's part of your hormonal communication topic area. So if you haven't already done so, do check out my video on the secretion of insulin. So I'm just going to touch on that very basically here. If there is a high blood glucose concentration, that is going to activate our beta cells to secrete insulin into the bloodstream. Now, that insulin is going to then bind to receptors on target cells. Now, this is virtually all of your body cells, but we're going to look specifically at the response that occurs at the liver and these body cells. So, if the insulin is going to bind, it binds to glycoprotein receptors. Now, these glycoprotein receptors are on the cell surface membrane. Therefore, we know that insulin is not able to cross the phospholipid bilayer. What happens when the insulin binds is those glycoproteins actually change their tertiary structure. They are proteins, therefore, it affects that tertiary structure in the R group interactions when the insulin binds. What then happens is these channels, these glucose channels, are now open. So glucose will then be able to be absorbed into the cells. So this will increase the absorption of glucose into the cell. And remember, that happens in all body cells. That increased absorption into cells is going to decrease the blood glucose levels. So what happens when the glucose actually gets into the cells is that the insulin also activates enzymes. And that activation of this enzyme results in what's known as glycogenesis. Now, this is where the glucose, the monomers, are going to be converted into the polysaccharide, the glycogen, through lots of condensation reactions. And we need to remember that our glycogen is our branched molecule 1 to 4 and 1 to 6, which means that we can have this rapid conversion of that glucose, binding it to the branches, therefore it becoming a stored molecule. So glycogen is this storage molecule for our glucose. What can also happen is the glucose can actually be converted into lipids, and that's through, again, this activation of enzymes resulting in what's known as lipogenesis. So genesis to make, lipo, lipids, glycogen. So the other thing that glucose can actually be used for is for respiration. So there's an increased respiration rate inside our cells, inside our liver cells. So what happens then if our blood sugar levels get too low? Well, this is where your pancreas, but this time instead of the beta cells, we're now looking at the alpha cells. So glucagon is a hormone which is released from the alpha cells. Now, I like to call it glucose-agon to remind me that it is glucagon which is going to be used when the blood sugar levels are too low. So low blood glucose concentration is detected this time by my alpha cells in the pancreas. They're going to respond by secreting glucagon into the bloodstream. So when the glucagon actually is transported in the bloodstream, it's going to, again, just like the insulin, bind to receptors at the liver. Now, this is important. This is at the liver, not at all body cells. So insulin has receptors in all body cells except your erythrocytes, whereas glucagon actually binds to receptors at the liver. That's going to cause the liver to break down, hydrolyze the glycogen stores, releasing glucose back into the bloodstream. So we are taking a low blood sugar level and we are raising it by putting glucose back into the bloodstream. So glycogenolysis, lysis to split. We're going to split the glycogen. We're going to break those 1 to 4 and 1 to 6 glycosidic bonds. It's a branched molecule. we can do that rapidly, therefore making it easier to release the glucose so that we can get our blood sugar levels back up to that level which is necessary. What else can happen? Well, again, if glucagon binds the receptors, it actually activates enzymes for what's known as gluconeogenesis. This is where there is a conversion of this time amino acids and glycerol into glucose so that that glucose can then be released into the bloodstream, raising our blood sugar levels. So it's important that we note whether we're talking about insulin from our beta cells or glucagon from our alpha cells, they are working antagonistically against each other. That is an example of negative feedback system. So the negative feedback system is the fact that there has been some form of stimulus, whether that is an increase in our blood sugar levels or a decrease in our blood sugar levels, and we are going to reverse that stimulus, negative feedback. We are going to reduce the magnitude of the stimulus by the response from that insulin that's been secreted by our beta cells or from the glucagon that's been secreted from our alpha cells. So insulin's response is in all cells, including the liver: increased glucose absorption, increased respiration, glycogenesis, lipogenesis. Glucagon response: glycogenolysis, reduced glucose absorption, reduced respiration, and gluconeogenesis. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. If you haven't already done so, do check out my revision platform, www.aiqchat.com.